Chapter 6. Maybe it couldn't be done, and that would solve all the problems. For decades, however, scientific notables had been predicting that clonal or vegetative reproduction would one day be practiced on humans, and it seems simple. Ordinarily, a new human is created by the union of an egg and sperm cells. Each contains half complement of the necessary number of chromosomes to chart a new life. They provide a full complement with millions of bits of genetic data intermingling to produce an individual who unites the hereditary endowment of his or her two donors. The offspring of this sexual unit is uniquely his own person in nature. In its tireless quest to keep its options opened, gambles on hybrid vigor, on the idea that its seed will, in these new combinations, be enriched and thus eventually go on to bigger and better things. It was a gamble that usually paid off, or so we like to believe, those of us who believed in evolution. For a long time, however, scientific seers spoke in their closed circles of another way of making men and women. It was all discussed in theory, but nobody could think of why it might not work. Except sex cells, the egg in the female and the sperm in the male. Nearly every cell in the body comes individually equipped with a full complement of chromosomes, copies of those that combined at the moment, sperm and egg, each with half its complement, as noted above, joined to create that body. In nearly every cell of the body, then, is every bit of information needed to recreate that body in its entirety. The main reason these individual cells do not multiply or try to in millions of bodies is that almost all of the genetic apparatus in each cell is switched off by biochemical suppressors. The only part of the total replicative blueprint that isn't blocked out in a skin cell, for example, is the tiny part that contains the instructions on how to make skin. Likewise, down the list for bone, hair, heart, nerve, etc. And if you remember when I was sharing from the book on Can Science Make a Human, that's exactly what it was talking about. It was hypothesized that if a very dexterous and clever technician, a master microsurgeon, really, could take the nucleus of a body cell, any body cell practically, and somehow get it undamaged into an egg cell whose nucleus had been removed or inactivated without damage, there would be an excellent chance that the total replicative machinery of the body cell would be switched on. And that would proceed to divide and differentiate, recreating, in a sense, the individual from whom it had been taken in the first place. If a person could be said to be a chip off the old block, that's this. This unique way of making people appealed to those who, however much they might respect evolution, nonetheless, thought that nature could use a little help now and then. Such persons spoke optimistically, even wistfully, of cloning great thinkers, scientists, athletes, heroes, and entertainers. Later, because many thought that members of a common clone might be able to communicate telepathically or at least intuitively, owing to their identical neurological schemata, some suggested cloning astronauts, underwater explorers, spies, and others who could benefit from unusual rapport in hazardous undertakings. Pessimists, on the other hand, thank their lucky stars cloning had not been available to Genghis Khan or Hitler. And in their writings and prognostications, they populated the future with ruthless armies of carbon-copied, single-minded doppelgangers under the direction of their despotic donors. The list of those who had taken cloning seriously was long and included distinguished men of science. For one, J.B. Haldane. Remember? He was also in the work that I just did. I just recorded yesterday about Haldane, who was in 1924, I think, one of the main people who recognized the ability to make parts of a man at that time. He was considered one of the most brilliant scientists of the century. Now listen to this. I went and looked his name up in a book written after the 80s. Not even there. And I was looking in a science and physics book. Yeah, I'm going to look again. Well, let me just do it right now. Haldane. I'm going to look it up in Asimov's Chronology of Science and Discovery and just look it up in the back, because I know that it should be in here, wouldn't you think? If he was the guy that figured out how to recreate creation. Okay, H-A-B, H-A-F, H-A-H, H-A-L, H-A-N. No Haldane. I thought that. I looked it up in another book yesterday, but you know why, don't you? We only know what we are, what they want us to know. I hate using the word they, but what can you say? Okay. Okay, so Haldane, I didn't know this, but it had been his idea to clone individuals with special effects, like night vision, a lack of a sense of pain, an inability to hear ultrasonic sounds that might be used in weaponry in the future. Did you hear that? Dwarfism, which he believed might come in handy in the high gravitational fields of some of the planets we might eventually need to explore to colonize, and so on. Another was Jean Rostand, the noted French biologist who thought cloning might be used to convey a sort of serial immortality for an individual by replacing worn-out copies of yourself with another indefinitely. I don't know, but I'm 71 and you get a little tired of working hard, and I'm not sure I want another go-round. Whatever, clever. Okay, and then other people that were interested were Nobel Prize winner Joshua Lederberg, doctor, who had suggested that neurological similarities of body cell donors and their clonal offspring might serve to bridge the generation gap. Interesting. Donors would be used to teach their clonal young. And then Dr. Carlson of UCLA proposed cloning the dead to bring back individuals of identical genotype. He was thinking we might be able to bring back King Tut. And then Dr. James Danieli who believed that multiple copies of a single individual placed in different environments would clear up many of the questions related to nature versus nurture controversy. And then Dr. Davis of the Harvard Medical School proposed that cloning talented individuals who might enormously enhance our culture. And Dr. James Watson, another Nobel Prize winner, saw in human cloning something akin to the collapse of Western civilization, but felt nonetheless that it would soon be achieved if it wasn't guarded against. It was just a lot of blue sky talk, however titillating or terrifying depending on one's outlook, up until the 1960s when Professor Stewart and colleagues at Cornell University took cells from the body of a carrot and placed them in a carefully concocted nutrient bath which contained things like coconut milk. In the solution, something remarkable happened. Some of the cells began dividing as if they had been pollinated, but they hadn't. Some of these switched on body cells not only divided, but gave rise to bumps and clumps that sprouted roots. In other nutritive mediums, they put up green shoots, and in soil, some of them developed into adult carrots, normal in every way. We were hardly prepared for the dramatic effects, Dr. Stewart said. It was as though the coconut milk medium, thought to be like those in the nutrient fluids of all plant embryos, apparently acted to engage the instructional data in the cell's nucleic acids in such a way that the cell's full potential for protein synthesis and thus for full growth was abruptly unlocked or disinhibited. So it was, in any event, that the 1902 prophecy of Austrian biologist C. Haberlandt that clonal reproduction would one day be achieved was fulfilled at last. The products of Dr. Stewart's ingenuity were called clones, and the process itself cloning. The root of this is the Greek word klon, K-L-O-N, which is variously said to mean twig or slip, cutting or throng. A clone is now regarded as a cluster of identical cells or organisms, or a single member of such a cluster or throng, all propagated from the same body cell. It wasn't long until the clones included animals as well as plant members upon the living universe, beginning in the late 1960s and building upon the pioneering work of the two American scientists, Dr. Briggs and Dr. T.L. King. Dr. J.B. Gurdon of Oxford repeatedly cloned the African clawed frog. He first destroyed the nuclei of an unfertilized frog egg with ultraviolet radiation. Now, that goes back to what I'm talking about radiation and the blue light. Then he microsurgically inserted into them the nuclei of frog body cells obtained from such remote places as the lining of the intestines. Some monitoring mechanism in an egg's cytoplasm, analogous to the white of a chicken's egg, noticed that there was a full set of chromosomes in its nucleus, and thereafter the mechanism behaved just as though the egg had come by its full set of chromosomes through fertilization by sperm. It instructed the chromosomes to divide and produce a whole new frog. In the wake of these startling experiments, noted Caltech biologist Dr. Robert Sinsheimer predicted in 1968 that it would be possible to clone a human in 10 years. Dr. Lederberg was simply optimistic or pessimistic, depending on how one interpreted his words. Dr. Atwood, a professor of microbiology at the University of Illinois, said about the same time that with a crash program, human cloning would be achieved almost immediately, but definitely within a few years. That's 1968. To many, the accomplishments of Stuart Gurdon and others showed that the major obstacles to human cloning had already been surmounted. It would be written in the New York Times Magazine that the Cornell carrot confronts our incredulity. To a scientific mind, the leap from single cell to cloned carrot is greater than the leap from cloned carrot to cloned man. Chapter 7. I spoke with Dr. Hirschborn, chief of the Division of Medical Genetics at Mount Sinai School of Medicine in New York. He served as American Society of Human Geneticists. Did he think cloning of humans was possible? Not only possible, but probable, he said. Human cloning was going to be more difficult than the cloning of carrots and toads, mainly because human eggs must be carried in the womb, not deposited on a rock or dropped into coconut milk. And human eggs are much smaller than frog eggs and much more fragile. The biggest challenge for those who would clone a man would be getting the tiny body cell nucleus into the egg-celled cytoplasm. But once you get the chromosomes from the body cell in there, there's no reason why the damn thing won't grow just like an ordinary fertilized egg cell, said Hirschhorn. As for getting the egg cell into the womb so that the clone might be nourished and grow, a technique had been worked out to accomplish this, and in fact had already been applied to a large number of animals. Reproductive biologists catering to animal breeders, for example, had recently learned how to cause prize cows to super ovulate by administering various hormones that induce mass masturbation and release of eggs from the ovaries. I remember this because I lived on the farm when I first got married, and my husband owned cows, and at that time they were dairy cows, and at that time artificial insemination was very popular, and the amounts of money, hundreds of thousands of dollars, were paid for specific ampules of semen, and the people would come by and artificially inseminate the cow, and if the cow was known to give a lot of milk or to be really healthy, etc., or specifically a bull, then you were set. You were just creating money, really. Okay, so the next step was the artificial insemination of these super ovulating cows with the sperm of prize bulls, creating in one cow dozens of super embryos. And then these were flushed by the fallopian tubes of the cows and transplanted one by one in the uteri of the other. Less exalted cows which, as mother surrogates, bore the burdens of pregnancy. In this way, a highly valued cow could be made to produce hundreds of calves without even having to go through a rigor of pregnancy. I remember this so well, and I have friends now that could really explain a lot more about this, which I might need to talk to. Now, doctors and researchers seeking to alleviate the anguish of childless couples were similarly making advances toward the goal of transplanting human embryos from one woman to another. A dozen or more researchers in several countries were regularly publishing results of their experiments. It was like a race was on, like the race to the moon. The first to succeed, it was expected, would reap overnight fame or notoriety if something went wrong and one of these test tube babies emerged mentally or physically defective. One plan was to remove the eggs from the ovaries or otherwise normal woman who was infertile because their fallopian tubes were blocked or had been destroyed. Their eggs would be fertilized in the test tube with the sperm of their husbands and then implanted into their own wombs. Another plan called for taking the egg of one woman, fertilizing it in a test tube, and then transplanting it into the womb of a second woman who could not produce her own eggs. Many objected to these experiments on the grounds of other things that deformed or retarded babies could result. Others protested that test tube fertilization was intrinsically immoral, regardless of any possible risk or benefit. Still others feared that such procedures would rapidly lead to a demand by many women for more frivolous and more dehumanizing uses of this new technology. Women who simply didn't want to undergo the difficulty of pregnancy could hire a woman to bear their children for a fee. One critic labeled this practice wombs for rent. Others said the poor would become the literal vessels of the rich. Still, the research continued for the good and humane reasons. Dr. Watson said that unexpectedly, there was rapid progress in the embryo transfer work, and it would pave the way for human cloning. This was made even further possible by additional breakthroughs in the area of cell fusion. Researchers would soon be able to join body cell nuclei to egg cell cytoplasm without even bothering with microsurgery. Chemicals were being found which would induce the two to fuse as if by magic. Keep a note on that. Chemicals found to induce the two to fuse as if by magic. Viruses were also being isolated that could achieve such fusion. Indeed, as one of those who opposed cloning, Watson worried that it might be unstoppable, given the same research advances required to bring it off were also those which would benefit mankind in important and diverse ways. Talking about a two-edged sword. Cell fusion techniques, for example, were promising one of the best avenues for understanding the genetic base for cancer. Already malignant cells were being fused with normal cells in an effort to isolate the individual chromosome that might induce specific forms of cancer. To stop these kinds of experiments would be problematic. I remember when they were talking about using stem cells, and I was so against it as a conservative Christian at that time. That was the rhetoric. And my best friend's son needed the stem cells to be pushed so that they could examine his disease better, and they were right on the cusp, and so it was really a two-edged sword. My values and principles, every one of them was challenged in trying to figure out what was right and wrong. How could you choose? It's like choosing one life over another, or I don't know. It's a really deep rabbit hole. But let's continue. Embryo implantation work that could provide new insight into contraception and even new solutions to the population problems would also see benefits that might outweigh possible risks. And so, says John, I found myself again confronted with should I or could I or I reread one passage I'd written a few years before with the advent of artificial wombs. And a couple of those articles were wombs, I reread one passage I had written a few years before in which I discussed speculation that with the advent of artificial wombs, and a couple were then already under development, clonists could forget about mother surrogates and instead merely deposit their body cell nuclei into these mechanical uteri and wait for their copies to emerge 9 months later. With the perspective of those few years and the revelations of Watergate washing all around me, I found myself no longer casual about the possible wonders of ectogenesis. Complete test tube pregnancy and babies decanted from bottles, as in Aldous Huxley's possible prophetic novel Brave New World. Babies exposed during those all-important formative 9 months to the whims and fancies of contemporary predestinators who just might, out there in the real world, have names like Ehrlichman, Haldeman, and Nixon. Dr. Glass, who holds the title of Distinguished Professor of Biology at the State University of New York, stated that ectogenesis will loom at a very real possibility by the end of the century. Mass cloning, he believes, might be difficult to resist. In an address that was later published, he worried about the temptation to replicate popular or commanding figures and envisioned a concealed clone spread through a population. Man, that really makes you think about. I was reading not that long ago about the fact that every president has at least 3 stand-ins. And then you think about how difficult it would be, but maybe not at all. And they say that the truth is harder to believe than any fiction. And can you imagine if some of the movie stars and the people that are leading us are clones? Okay, popular demand could result in clones of such celebrated individuals as John Kennedy or Dustin Hoffman or Elvis popping up all over, provided someone had procured some of their living cells. Something not difficult to do. A piece of skin could suddenly be worth a fortune, or the cloning of a black market. The mind boggles at the bizarre possibilities. Glass warned the use of cloning by tyrannical governments. Such regimes, he said, the production of human clones might have an irresistible advantage for. Another scientist, Lord Rothschild, recently warned a gathering of scientists that self-centered fanatics in the Hitler mold might be tempted to set up shop privately, making dozens of replicas by themselves on the sly before society could intervene. Dr. Hirschhorn had also warned me about the possibility of do-it-yourself clonists. Lord Rothschild was so concerned on this score that he had proposed the establishment of a commission for genetical control to license clonists, at least implying that in his view, cloning might be justified under some circumstances. Dr. Lederberg, who sometimes seemed to be enamored at the concept and other times contemptuous about it, sometimes even bored by it, expressed that where tribalism and racism had prevailed, clonism and clonishness would assert themselves in the future. The ethnocentrism of clones, embracing perhaps dozens of thousands of identical individuals, would be formidable, far outstripping mere clannishness or cliquishness. Each clone would consist of members of only one sex. They might become so insulated that they would not even mix with the opposite sex. They wouldn't have to, at any rate, because they could go on indefinitely, reproducing themselves asexually. The female clones possessed of both wombs and egg cells would have some advantage at the onset, but the male clones would use slave females, or more likely artificial wombs. It was not too preposterous, some claimed, to envision a massive war of the clones sometime in the future. Though these fears and concerns had to be taken into account, many of them struck me as fanciful and unfairly pessimistic. I felt that some of the critics of cloning were not entirely forthright in making their arguments. James Watson took positions and then sometimes stepped aside from them. In the Atlantic Monthly, he contended that the first reaction of most people to the cloning of a human being would be one of despair. The nature of the child-parent bond, not to mention everyone's values about their individual uniqueness, could be changed beyond recognition. To people with strong religious backgrounds, our most sensible course of action would be to de-emphasize any research that would circumvent the normal sexual productive process. He added, cell fusion experiments might no longer be supported by federal funds. Even more effective would be to take steps to make illegal or to reaffirm the illegality of any experimental work with human embryos. This was difficult to deal with because you couldn't tell whether Watson was directly proposing to make it illegal or to deny funding to such research. He used as a cover, if you were a person of strong religious background. Was he such a person? I was concerned about cloning, but annoyed at what I regarded as Watson's hit-and-run attitude. I wish he had better expressed what he thought and felt. I wanted guidance, untainted by scientific politics, and I believed that Watson was not entirely sincere. He seemed more interested in taking down a few notches of his British colleagues rather than forming any constructive ethical position. Later on, I'd be amused and relieved to hear Watson testily describe as kooks and shits and incompetents the critics of recombinant DNA experiments, which nearly everyone in the genetic engineering field readily conceded to be potentially far more perilous than embryo transfers or cloning. Watson was deeply involved in the new work, but had never had a hand in the embryo or cloning research. As I reviewed everything I heard or could find on this subject, nearly everyone who was in a position to know believed that human cloning was a very real possibility, and all that was needed was the available talent with money and will. Someone had to want to do this and want it badly. I knew now that that existed. But many of those who believed that it could be done believe strongly that it should not be done, at least not yet. Why yet? Still, there were those, and among them some famous scientists, who had seen fit to point out the positive possibilities. Lederberg himself observed that it would make possible unrestricted duplication of prized cattle, horses, animals important to men. Further, in his words, human cloning could facilitate the free exchange of organ transplants between cloned individuals, with no concern for graft rejection, no concern since the cells of one member of the clone would be identical to the other in every way, and thus entirely histocompatible with those of any other member. The ability to make valuable duplications of celebrated individuals had been noted and argued as an incalculable amount of basic or pure scientific data would accrue in the process of learning how to clone a man. Data related to cell differentiation, cancer, genetics, immunology, and as others pointed out, at the wake of successful cloning, psychological, neurological, and other consciousness-oriented research might be substantially aided by the availability of multiple identicals. There were other issues. Those men of religion and philosophies are the heavies. Then came a startling report from New York, which dramatically catalyzed the making of my decision. Chapter 8. A doctor, which I had worked with in the writing of a book and whose work I knew well, called me to relate a disastrous series of events. Landron Shettles was his name. He had been a distinguished career as an attending physician, researcher, and professor at Columbia Presbyterian Medical Center and at the Columbia University College of Physicians and Surgeons in New York City. Now, following an explosive incident, he had to resign. He told me that his superior, Columbia Presbyterian, Dr. Wheel, had purposely destroyed a test tube specimen containing, he said, the sperm and egg of a Florida dentist and his wife, a patient of Dr. William Sweeney. Shettles believed that he had achieved a test tube conception, that the sperm had fertilized the egg, and that the resulting embryo was incubating and could shortly be implanted into the uterus of the wife, whose infertility had been caused by irreparably damaged fallopian tubes. Shettles was preparing, in cooperation with Dr. Sweeney, a clinical professor of obstetrics and gynecology at Cornell, to carry out what might have proved a history-making operation when he found the specimen had been confiscated. the sterile conditions under which it was being maintained broken, and the entire attempt terminated by Dr. Weil. He added that he was called on the carpet and that Dr. Weil became emotional and asked him if he was trying to create a monster. He was then accused by his superior of breaking the law by attempting this procedure and was ordered out of the state hospital at once, or out of the hospital, not state hospital. He noted that the lock on the lab where the eggs were incubating was immediately changed and a guard placed at the door. In the uproar that ensued, attorneys for both sides agreed that Dr. Shettles would resign voluntarily and Dr. Weil and others at Columbia would refrain from making any other critical remarks and refused to comment, except to say that Shettles had decided to resign. The Florida couple, Doris Delzio and her husband, filed a $1.5 million lawsuit against Weil in federal court, claiming that termination of the implant procedure without their consent denied them their last opportunity to have a child. That litigation was in 1978 and was still unresolved as of this book. I was hurt and saddened by what this might do to a man whose work I had written about on several occasions and whose humanity and good intentions couldn't be questioned. I could not believe this had happened to a man of his stature. He had received his MD and PhD at Johns Hopkins. His historical microphotographs of his earliest stages of human life from conception onward have been included in more than 50 textbooks and displayed in the American Museum of Natural History in New York and the Academy of Sciences in Moscow. Dr. Rock, one of the principal developers of the birth control pill, had hailed Shettles' test tube fertilization of human eggs and his culturing and study of embryos as a landmark in our insight into human embryogenetics. Dr. Guttmacher, while serving as national president of Planned Parenthood World Population, said he was endowed with an ingenious mind and superb technical ability. And Frederick Heder, son of the late U.S. Secretary of State and noted surgeon at Columbia, said that his productivity in the laboratory had been prodigious. He's known the world over for his investigations into the mechanisms of conception and developmentation of early fetal life. If a giant in the field like Shettles could be shot down for merely making preparations to implant an egg removed from a woman back into that same woman, it was evident that I must be flirting with an area of science that was far more controversial than I had ever imagined. There seemed to be a feud involving professional jealousy between Shettles and Weil that had precipitated the event, and the procedure in question had given Weil the ammunition he was looking for to knock off a man whose reputation exceeded his own. Boy, is that a whole topic of conversation. Scientists and funding and egos, and how that affects you and me. He granted me an interview in the course of my writing an article which would appear in New York Times. Weil claimed that clearance for the procedure had never been obtained by Shettles, and what he did was illegal and immoral. Dr. Shettles maintained that no clearance was required, since Mrs. Delzio was Dr. Sweeney's patient and the actual implant operation would be performed at another hospital. Only in the in vitro fertilization phase would take place at Columbia. As for the alleged illegality of the act, Dr. Shettles characterized this as a slander. and asserted that no law existed to prohibit his work. Blah, 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 blah. Okay, so you get the point there. Okay, so the morality was the crux of the problem. Who is going to get permission for the fetus? What he meant was, who said that if the fetus could not grant permission for its own test tube conception, then it was immoral to create it, because in the act of creating it, there was the possibility of creating something monstrous, retarded, or deformed. One should not take lightly the possibility of producing damaged babies through laboratory manipulation. It would indeed be immoral to proceed if the risks seem substantial. But scientists believe that there was sufficient animal data already attesting to the ability of man to manipulate germinal material and still get healthy offspring. I mentioned experiments in which an entire herd of cattle embryos, for example, was transported across the Atlantic, incubating in the uterus of a live rabbit, being extracted and successfully implanted in foster mothers, which gave birth to normal calves. This was all done because it was cheaper to send the embryos frozen inside surrogate mother. Crazy stuff. When fetuses were afflicted with gross abnormalities, nature almost always spontaneously aborted them. Some of the abnormalities could now be detected with prenatal diagnostic techniques and instruments. Didn't the risks sometimes justify the ends, alleviating the anguish of those who were childless? Wheel said the issues were more complex than anyone could imagine. Next, he debated in Chapter 9 with Dr. Cass. Dr. Cass was not the principal opponent of embryo transfers and cloning in the world. He was the most vocal. In one of his papers, he described cloning as the contrived perpetuation into another generation of an already existing genotype. It was his view that the last obstacles to cloning were about to fall by the way. The fusion experiments by Dr. Graham at Oxford, the in vitro fertilization and embryo transfer work of others were advancing at such a pace, he said, that it's reasonable to expect the birth of the first cloned mammal sometime in the next few years. Again, it could be done. And now we know about Polly. Is it Molly? It's Molly the sheep, right? So here's a laundry list of cloning applications that apologists and titillator's provided. I'm trying to skip the boring parts here. Replication of individuals of great genius or great beauty to improve the species or to make life more pleasant. 2. Replication of the healthy to bypass the risk of genetic disease contained in the lottery of sexual recombination. 3. Provision of large sets of genetically identical humans for the scientific studies on the relative importance of nature and nurture for various aspects of human performance. 4. Provision of a child to an infertile couple. 5. Provision of a child with a genotype of one's own choosing, of someone famous, of a departed loved one, of one's own spouse, or oneself. 6. Control of the sex of future children. 7. The production of sets of identical persons to perform special occupations in peace and war, not excluding espionage. 8. Production of embryonic replicas of each person to be frozen away until needed as a source of organs for transplant to their genetically identical twin. 9. To beat the Russians and the Chinese, to prevent a cloning gap.